In this part of our series on adaptive immunity, we're going to discuss the effector functions of the T cell response. So we left off previously discussing that T cells get activated within lymph nodes. Those T cells can develop a number of specialized effector functions depending on the inflammatory environment in which they get activated. This includes the development of helper T cell subsets, including Th1 cells, which primarily can respond to bacteria, viruses, fungi, and even single cell parasites. Th2 cells, which primarily respond to helminths, but also participate in the allergic response. And Th17 cells, which are important T cells in the response to extracellular bacteria and fungi and are important for maintaining barrier integrity at mucosal surfaces. On the flip side, CD8 positive T cells differentiate into killer T cells referred to as cytotoxic T lymphocytes and are primarily important in the antiviral response. As a reminder, all of these activation and differentiation events occur in the T cell zone. So naive T cells get activated and become Th1 or Th2 or Th17 cells and migrate to inflamed tissues. Some T cells stay in the lymph node and migrate to the B cell zones to help B cell activation. The first thing I want to note is that when activated T cells migrate to inflamed tissues, they do so via very similar mechanisms as neutrophils. And so I'm not going to talk about it much, but for example, they express the high affinity integrin LFA1 to recognize and attach to activated endothelial cells, squeeze through tight junctions, and migrate to sites of inflammation. Th1 cells, once they migrate to sites of inflammation, interact with activated macrophages, and when they interact with those macrophages, produce a cytokine called interferon gamma. Interferon gamma is an important cytokine that activates macrophages. What do I mean by this? Well, activated macrophages have enhanced phagolysosome function and enhanced cytokine production, and thus are much better at eliminating intracellular pathogens within their phagolysosome. And so this is an important mechanism whereby macrophages are able to eliminate pathogens that might have been able to initially resist the effects of the phagolysosome. Th2 cells, when they migrate to tissues, are important in activating certain types of immune cells, such as eosinophils and mast cells. And they do so through the production of three cytokines: interleukin-4, interleukin-5, and interleukin-13. These cytokines induce degranulation of eosinophils and mast cells and are important in the anti-helminth response. They also participate in the allergic response, and they enhance macrophage tissue repair functions. These three cytokines are also important in promoting the production of IgE antibodies in response to exposure to things like allergens. This can lead to mast cell activation and the allergic response, which is also known as a type 1 hypersensitivity response. Th17 cells produce a couple of cytokines. Interleukin-17 mediates the recruitment of neutrophils into inflamed tissues, and IL-22 can act on epithelial cells and mucosal surfaces to enhance barrier function. This includes the increased production of mucus and antimicrobial molecules like defensins and lysozymes. Th17 cells are abundant in the gut. Lastly, there is a subset of T cells called regulatory T cells, and these regulatory T cells are immunosuppressive. They can suppress the activity of antigen-presenting cells. They can suppress T cells directly. They make immunosuppressive cytokines to do that. They can arise during thymic development, but they can also be induced during an immune response. And lastly, CD8 positive T cells can differentiate into cytotoxic T lymphocytes, and CTLs can directly kill virally infected cells. They form a cytoplasmic bridge with their target and poke holes in their target membrane. They are then able to deliver a granule payload into the target cell that induces apoptosis